So hello and welcome to this episode of Our Longevity Futures. I'm Chris Curwen, your co-host, alongside my good friend and colleague Antonia Schulze-Moscow. And today we are delighted to be joined by Professor Matt Kaeberlein. So Matt is a professor at the University of Washington, where his research focuses on the basic mechanisms of aging. He's also the co-director of the Dog Aging Project in Washington, which we'll be talking more about today. So welcome, Matt, and thank you very much for joining us.
Thank you. It's a pleasure to be here.
So we'll start off by asking you a little bit about your background. So how did you actually get into the world of longevity and the science that you're researching today?
Yeah, so I mean, it's actually it was an accident, to be honest with you. I went to graduate school at MIT, thinking I was going to do structural biology or X-ray crystallography or something like that. My undergraduate training was in biochemistry and mathematics, so very sort of biophysical. And I heard a talk by Lenny Guarente, who I ended up doing my PhD thesis with during my first semester at MIT, where he talked about how his lab was using genetics and molecular biology and biochemistry to study the biological aging process. It was in yeast at the time. And, I still to this day, I don't exactly know what it was about that just captured my interest and enthusiasm, but I just really got turned on by the idea that you could scientifically study something as complicated as the biological aging process. Now, as I said, that was in yeast. And, looking back, to be completely honest, I have to admit, at the time, I didn't even care whether what we were studying was going to be relevant for human aging. I just thought it was such a cool and complicated, biological problem that I wanted to dive into it. That aspect of my view of aging has changed over the years, where now I'm very much interested in what we can learn about human aging. But I think it was really just the complexity and the challenge, and the fact that I recognized even at that time, that aging is really one of the unsolved mysteries of biology, and so to be able to have the potential to have an impact there, I thought would be really cool.
Yeah, I think your story sums up so many people's journey into longevity, just the sheer curiosity and interest in it.
Yeah.
But as I mentioned, you're the co-director of the Dog Aging Project, which is something that every time I look into it, it just fascinates me and just strikes me how genius and well run it is. So can you just tell me a bit more about what the Dog Aging Project is and how it was actually started?
Sure. So the Dog Aging Project, really at its core is about trying to understand the biology of aging in companion dogs or pet dogs. And there's really two parts to it. One is a large-scale longitudinal study of aging. It's completely observational. And the goal there is to really identify the most important genetic and environmental factors that influence healthy aging in companion dogs. The second piece of that is to actually try to impact the biology of aging in companion dogs through clinical trials, testing interventions that we think, based on laboratory studies, might actually slow aging or in some cases reverse aspects of aging. And so right now we have one clinical trial. It's a clinical trial of a drug called Rapamycin. But I think my vision for the future is that our studies won't be limited to Rapamycin, and in fact, the goal there is actually to have an impact on aging in pet dogs. I think what fundamentally differentiates the Dog Aging Project from most of the non-human research in the field is the fact that these are actual companion animals living in the real world, right? And so that's, other than studies that are carried out in humans, there really aren't any studies in the field that, experience aging in the real world. And so I think that's one, sort of unique aspect to the Dog Aging Project that from a scientific perspective is really important.
So to jump into the biology a bit, one of the most well-known things about dog aging is that typically smaller dogs outlive larger dogs. So Why exactly is this?
Yeah. So before I jump into that point, I actually think this raises a sort of even more fundamental point that I want to make. And another reason why I really like the Dog Aging Project is I think that dogs in particular are a really powerful animal to illustrate the idea of biological aging to the general public. I still think most people, if you just walk up to somebody on the street and you start talking to them about the biology of aging, they're going to have no clue what you're talking about. And I think dogs are a really useful sort of example that people understand. We've all understood since we were kids this idea that 1 human year is about 7 dog years, right? That's not perfect, but it's pretty close. And if you just explain to people what that means, right? Another way of saying that is dogs age about 7 times faster than we do, right? Than humans do. And so that tells you immediately that there's something about the biology of dogs that's different from people, that regulates that rate of aging. And so if we can understand that biology, we have the potential to actually have an impact on that biology to modify the rate of aging. So I really like dogs for that reason, because this idea of human years and dog years is exactly what we all talk about in this field all the time, that aging is biology and can be modified. We just have to have ways to communicate that to the general public. And I think dogs are a really great example of that. So that wasn't what you asked, but I wanted to share that because I think it's a really powerful sort of message to get across. So you asked, why is it that big dogs seem to age faster than small dogs? Certainly big dogs have shorter lifespans or life expectancies than small dogs. That's well established. And it seems like they are aging biologically more rapidly than small dogs are. And you can see that, based on functional declines. So changes in heart function or arthritis or kidney function are all accelerated in big dogs. They also have higher risk of cancer pretty much at every age, chronological age. A big dog has a higher risk of almost all forms of cancer than a small dog does. So it seems like biologically they're aging more rapidly. I mean, I think we have to be honest. We don't know with certainty why that is, but there's a very plausible hypothesis that is suggested based on the genetics of body size in dogs. So the most important genetic variant for affecting body size in dogs is in the IGF-1 pathway, insulin-like growth factor 1 pathway. Elaine Ostrander showed this many years ago in a very nice paper published in Science. So if you have a variant that increases IGF-1 signaling, you get bigger dogs. And of course, bigger dogs are shorter lived. Now, why that's interesting is we've learned from laboratory studies in mice and flies and C. elegans, worms, that mutations in the IGF-1 like signaling pathway that reduce signaling also increase lifespan. So that the directional arrows are the same in C. elegans and fruit flies and mice and companion dogs, that mutations that reduce IGF-1 signaling are associated and causal, at least in the laboratory animals, for longer lifespan. So it's a very plausible hypothesis, I think, that the same thing is accounting for differences in life expectancy associated with body size in dogs. Again, I'm a little bit careful to say that hasn't been proven because nobody's obviously done the experiment where you somehow maybe with CRISPR change IGF-1 signaling in a big dog and see whether you affect lifespan. But I think that it's pretty likely that that's the mechanism that's underlying that.
So we see
Can I jump in there?
Species. Oh, sorry, Jared, you go ahead.
Yeah, sorry. I just want to, on the wildcard question side. So one thing which is interesting though is wolves are much longer lived than dogs, if I'm right there. But wolves are similar size or more similar in size to bigger dogs. So do you think that's a separate pathway, or is it because wolves and dogs have diverged so much?
So I'm not sure it's actually the case that wolves are substantially longer lived than companion dogs. I mean, it's a little bit challenging, right? Because wolves, of course, are in a different environment, and anytime you're in the natural environment for an animal, usually lifespans are actually depressed, right? Because of things like predation, or if an animal gets injured, it's unlikely to survive the next winter, things like that. So first of all, that would be the first thing I would say. I would also say it's complicated, right? So companion dogs, while they have many powerful features for studying things like aging or other aspects of biology, they're also a little bit unique, right? In the sense, particularly if you look in purebred dogs are obviously relatively inbred. And so there is a depression in life expectancy that goes along with being purebred. If you just match the body size, purebred versus mixed breed, right? Probably because of that inbreeding factor. It's about a year at any given weight. It's about a year difference in life expectancy just because presumably of that inbreeding factor. So it's a little bit hard, I think, to draw, really apples to apples comparisons there. But I'm honestly not familiar with the data that suggests with any level of certainty that if you took a wolf or enough wolves in captivity and did a side-by-side sort of lifespan normalized to body size, that the wolves would live substantially longer.
So we've successfully studied and shifted the rate of aging in a number of different model organisms, namely yeast, worms, flies, mice. So how is the dog aging project actually different from these investigations that we've done previously?
Right. So, I mean, I already talked about the fact that in the dog aging project, these are companion dogs living with their owners in the real world. So that, I mean, is, I think, arguably the biggest difference between, the studies that have happened in yeast and worms and flies and mice. Those are all in the laboratory. And of course, in the laboratory, we are typically maintaining these animals in a highly controlled environment. It's relatively sterile compared to the real world, usually with an overabundance of food, right? So they are developing rapidly, selected for very high rates of reproduction in the laboratory. So it's just a fundamentally different experimental paradigm. And there's good reason why we do that in the lab, right? We're trying to control everything that we can and change only one thing in principle and see the effect of that one thing. But of course, humans don't live in a laboratory, right? And so I think the dogs, in many ways, companion dogs do a really good job of capturing the human environment, with the exception of diet. And we can talk about diet if you'd like. Diet is an interesting difference, of course, between companion dogs and people. But with the exception of diet, they really do share our environment in almost every way. And in some ways, they're exposed to aspects of the environment even more than people are, like drinking water, right? Most people don't give their dogs bottled water, they give them tap water, right? So it's really powerful from that perspective. So that's a really, I think, important and unique feature of the dog aging project. Another one is the genetic architecture that we sort of have already touched on, that you have a genetic diversity in dogs that's actually greater than the genetic diversity in people, but it's also in the context of this unique architecture where you have a few hundred purebred breeds of dogs, which in some ways are kind of like inbred strains of mice. They're not quite as inbred as like a C57 black 6 mouse, but they're pretty inbred. But then on top of that, you have the mixed breed population. So you really do have a heterogeneous genetic population of mixed breed dogs. So I think that's also a really interesting and important aspect of the dog aging project that, again, it's really hard to capture in laboratory animals. Very rarely do people do studies in more than one strain background. And when they do, it's usually only a handful, right? So this is actually a criticism people often raise about preclinical literature from the aging field, is that it's mostly comes from, a relatively small number of genetic backgrounds in each of the model organisms. And so, it's a valid question. If you were to take something like caloric restriction and apply it across a panel of genetic backgrounds, would it work as well? And the answer with caloric restriction is clearly no. In the studies that have been done, what we found is that about a third of genetic backgrounds in each model organism show no response or a negative response to the same level of caloric restriction. So, I think it's an open question whether some of these interventions that we study are going to be robust across a diversity of genetic backgrounds. And we can start to address that in a diverse population. And of course, and one of the goals is to translate to humans, that genetic diversity and environmental diversity is really important to understand how much of an impact it's going to have. So I think that's another area where the dog aging project is unique. And of course, it has the power that dogs age, about 7 to 10 times faster than people do. So we can't do studies in quite the same time frame as mice, but it's not so much longer, right? It only takes about 2 times longer really to do studies in dogs as it does in laboratory mice. And so you can do observational, correlational, or clinical trial studies in a feasible time frame in companion animals, which is that's the real challenge with trying to study aging directly in humans. Humans age so slowly that it would be a 20 to 30 year clinical trial to try to test whether something like rapamycin slows aging in a healthy human population, right? If lifespan was your endpoint.
Well, you very well summed up all the points which I really fell in love with what you're doing on the dog aging project.
Yeah.
And obviously,
Well, and I mean, I think, for me also, people love dogs. Like, I'm a dog person. I've had dogs my entire life. I've got a German Shepherd named Dobby right now.
I think I've seen the picture. Yeah.
Yeah.
Really
Who I love dearly. Right.
Yeah.
So, I mean, it's not only about the science and what we're going to learn about human aging. It's about, what we can learn about aging in dogs and potentially increase the healthy longevity of companion animals. So there is that aspect of it, which I think it just depends on your perspective. Some people, and that's partly why this project, I think, is so appealing to so many people, right? The people who want to have an impact on human aging recognize that we can learn something about human aging from studying it in companion dogs. And the people who love animals, love their pets, also recognize that this is important and potentially valuable for them. So I think it appeals to a pretty wide audience. And again, I also keep coming back to the potential, how can we message the field to the general public? And I think that's a really important aspect of the Dog Aging Project, that not only do people understand that their pets age more rapidly than they do, but they see the value. In some ways, it's easier for them to see the value if we talk about increasing health span and lifespan in animals and their pets, compared to if we start talking about trying to do it in humans. So I think it appeals to the general public in a way that really not very many scientific projects do.
I know at least one person on this call that would rather increase dog longevity than human longevity anyway. So I think a lot of people would be in that same boat. But when we talk about aging, I mean, it's well known in science, before you start to change something, you first have to measure it. So how do you actually measure biological aging in your dogs? And what sort of endpoints are you using?
It's a great question. And I think, this is a bigger question for the field, right? This is something that the field as a whole, I think, is working through, struggling with, trying to figure out what is a consensus definition for biological aging. Okay, so I think it's not an easy question to answer in a concise way. I think one view, and I actually don't disagree with this view. I would say this is my preferred view. I think we have to look at lifespan. There is a component of the field that says that, health span is more important, so we should look at health span and not worry about lifespan. I think we have to look at lifespan. Number one, because if you've really impacted the biology of aging, you will, by definition, impact lifespan. And number two, it's really the most quantitative measure that we have. All of the measures of health span are, at least overall health span, are qualitative. There is no single quantitative assessment of health span that the field agrees on. So when people, and this is a little bit of my pet peeve, and I will admit I have done this, I don't do it anymore. When you see in the title of a paper somebody claim that they have extended health span in an animal, they haven't. And reviewers should stop letting people put that in their titles or abstracts. You can't claim that you have significantly increased something you can't measure, right? So because there is no quantitative way to measure health span, you cannot claim that you have extended health span. So sorry, that's a little bit of an aside, but I think it's an important one for people to start appreciating. Okay, so how do we measure aging aside from lifespan? And I think there are different camps, right? I am, for now, at least in the functional camp. I like functional measures of aging. So looking at quantitative assessments of organ, tissue, cellular function. So for example, by echocardiogram, looking at function of the chambers of the heart, right? Or by blood chemistry, looking at function of the kidney and the liver and things like that. Or by strength tests, looking at function of muscle or activity measurements, right? I think that, again, and look, I was trained as a molecular biologist, geneticist, biochemist, right? I understand why people gravitate towards the molecular measures, but I think if your interest is really in quality of life, you really need to look at function. And again, I see too many times the basic scientists in this field say, oh, I've changed the epigenetic clock and reversed aging. No, you haven't. You've reversed epigenetic changes that go along with aging. You really have to show that functionally you have had an impact on aging. So I like functional measures. So in the Rapamycin trial, for example, we're looking at echocardiogram for heart function. We're looking at neurological exams for neurological function, cognitive assessments for cognitive function, activity monitoring, kidney function by blood chemistry. So we are trying to, as broadly as possible, look at functional measures of aging. Of course, we're also looking at disease incidence. So what's the frequency of cancer, kidney disease, diabetes, things like that in the dogs. And then we are also collecting the molecular measures, epigenetic clocks, metabolome, microbiome, but I view those as much more exploratory. Again, I'll say it again, if I see in dogs that rapamycin reverses or doesn't do anything to the epigenetic clock, let's say there's no change in the epigenetic clock, but the dogs are living longer and healthier, I'm going to believe the lifespan and health span metrics over the epigenetic clock. So I think it's really important to look at functional measures. If you want to look at the molecular measures too, great. But I don't view those as anywhere near as definitive as lifespan and functional measures of health. So those are the kinds of things that we're looking at in the clinical trial. In the longitudinal study, we're collecting pretty much every kind of data that you could imagine on environment, also genome. So we're getting genome sequencing on 10,000 dogs. And then I mentioned epigenome, metabolome, microbiome. So we're also doing the omics. But again, the longitudinal study is much more exploratory and observational. At this point, we don't know what the signatures are going to be that are predictive for future health outcomes. That's part of the point of doing the study.
So I know Jared is. I'm sure Jared's got a question at the moment, or Jared, I can wait till the end if you'd rather. But if you had a question now, go for it.
I'm just very tempted to ask more about.
Please do.
Matthew of the clocks. What you're saying there is kind of taking some stance, I guess, on the role of causality in those clocks. I just wonder if you had like an intuition, because
Sure.
They do correlate really well with age. Obviously, there are limitations. There's only measuring so many methylation sites. But if you had to take one side or the other, the clocks are a byproduct of some other process, or the clocks are measuring aging itself.
Yeah.
Where do you come down?
Yeah, I think so. My view is we don't know yet, and you're referring specifically to the epigenetic clock. So I think big picture, I have no doubt that we can develop molecular clocks that are predictive for biological aging. Specifically, are the epigenetic clocks alone going to be predictive for biological aging? First of all, I'll say I don't know. My intuition is that they will not in many situations. So I think that you can definitely find specific situations where a specific epigenetic clock is predictive, absolutely, for chronological age. That's unquestioned. And you can find individuals who fall off of that prediction for chronological age, who then are hypothesized to be biologically aging more rapidly or more slowly. And you can find evidence to support that, right, under those very specific conditions. I haven't seen anything yet that convinces me that in a general population, environmentally, genetically heterogeneous, changing over time, that the epigenetic clocks are actually predictive for individual future health outcomes, which is what we want, right? If the clock really works, you should be able to take it, you individually, get an answer, change your lifestyle, and see whether the clock is actually going to do a good job of predicting your future health outcomes. Again, part of the challenge with proving that in people is that people live so long, right? So it's hard to do those studies in advance, right? So what people have relied on are longitudinal data sets, which are, there are problems with doing that, as we all recognize. But you could do it in mice, and nobody's done it. And I actually think this is a real limitation to the clock field, where the clock people need to actually do the right experiment here. There is absolutely no reason nobody has taken a population of old mice, measured their whatever clock you want to measure, treat half of them with rapamycin or whatever your intervention is, and then at the individual level, show us the clock is predictive for outcomes, right? That's a doable experiment. It shouldn't take more than 2 years. And so, I feel like this is a case where people who are invested in these clocks have kind of skipped over actually proving that they work and started selling them to other people, right? And I just think it's a problem. I think it's wrong. Look, I get the commercial side of it. Like, I don't have any problem with people within the rules that the FDA has set up, starting businesses and making money. I do have a problem with scientists doing that. Personally, I don't think you can call yourself a scientist and then go start selling stuff that you haven't actually tested to people. I just think you shouldn't do that. So, and we see that in this field. It's a little bit unfortunate. The other concern I have is when the same people selling the clocks are also selling the supplements that supposedly affect the clock. I worry a little bit about that sort of conflict of interest. But I do think in general that clocks, we will be able to develop biomarkers. I mean, again, clocks are just kind of a sexy word for biomarkers, but this is something the field's been working on, since the 80s, right? 1980s. I think we will be able to develop diagnostic predictive biomarkers for future health outcomes during aging, probably not only involving the epigenetic clocks, but involving other parameters, blood chemistry, functional measures, maybe imaging of the face, right? There's people working in all of those areas. My gut feeling is all of those together will give you a more precise predictive algorithm than only the epigenetics. The other thing to say about epigenetics is, it's a little bit of an unknown. There are some people who kind of feel like the epigenetics are upstream of everything else in biological aging. Again, my intuition is that's not going to be the case, and that epigenetics are only monitoring one aspect of aging. Maybe it's a big aspect, but I don't think that the epigenetic changes are everything that's happening in biological aging. And time will tell. There's a lot of enthusiasm and excitement about the reprogramming idea. If it is the case that the epigenetic changes with aging are everything, then it should, in theory, be possible to, through periodic reprogramming, to make a mouse that lives, I don't want to say forever, but a long time, right? At least double the normal lifespan of a mouse. And again, this is where I think the hype has gotten way, way ahead of the actual data. Nobody's actually done it yet, right? Nobody's actually shown you can increase lifespan by 30% in a mouse with reprogramming, let alone double the lifespan. So, I feel like people should just do the experiment instead of getting ahead of themselves and making bold claims without any actual data, or much actual data, I should say.
There we go. Well, yeah, very well put, and a very poignant point, I think, at the moment in today's climate in the longevity field. But coming back to the interventions in the dog aging trial. So rapamycin is one of the interventions that you're using at the moment, and it's one of the most highly anticipated longevity interventions out there. So firstly, can you just give us a brief explanation of what rapamycin is? I know you must have done this 1,000 times, but for those people who haven't heard before.
Sure. Yeah. So I'll definitely tell you about rapamycin, but I want to comment on something you said. That's one of the most highly anticipated, because it's really interesting to me, actually, having been, working on mTOR and rapamycin since, I think we first started really studying mTOR around 2003, right? This was from an unbiased screen in yeast. So having been sort of around this pathway in the aging field for that amount of time, it's actually kind of interesting the way this evolved. So, we found, and then a couple other labs in parallel found in yeast and worms, that if you inhibit mTOR, you can increase lifespan. Nobody really paid much attention in the field until the interventions testing program showed in 2009 that you could start treating mice with rapamycin in middle age and extend lifespan. And then people got really excited for about two years. And then everybody got tired of rapamycin and got, interested in the newest shiny object, and nobody would pay attention. I mean, believe it or not, it was really, really hard to get any funding to study mTOR or rapamycin in aging, because the field kind of felt like, oh, we already know about that, right? And it's only been in the last, 2 years, I would say, that I have really felt an uptick in interest in rapamycin again. So, this happens in science sometimes. There's, this pendulum that swings, but and I think we still don't know ultimately, obviously, how useful rapamycin is going to be for lifespan and health span. But there is a noticeable uptick of interest in mTOR and rapamycin in the last 2 or 3 years in the field. And honestly, I'm not really sure exactly what's driving that, except I think we're starting to get hints that it could have some effects in people. And I think also the publicity that we've gotten through the Dog Aging Project has helped, because somebody is actually testing something in the real world, which I think people appreciate. So it's just interesting to me how the interest level has gone up and down.
It's like the fashion trend, isn't it? They come around.
Yeah, and I mean, this happens in every scientific field, although I think the aging field is more prone to it. There is definitely a shiny object syndrome in this field where people get completely fixated on, something. Right now it's reprogramming. It was senolytics a few years ago. And
I was about to say, yeah.
That's okay. It happens. I think ultimately, some things stand the test of time. some things don't, right? And unfortunately, sometimes it takes a long time to figure out, what actually has an impact and what was, where the hype got a little bit ahead of the actual science. So in any case, that was a little bit of a tangent, but so rapamycin, it's sort of an interesting molecule. It's produced by a bacterium that was identified on Easter Island, also known as Rapa Nui. That's where the drug gets its name from, rapamycin. So it's produced by this bacteria called Streptomyces hygroscopicus. It was identified in soil samples there. It had sort of a tortured history, and I don't have time, and I don't know that I could actually do a credible job of telling you the whole backstory, but it's a really interesting backstory.
We've got an article on our site which goes into it. It's one of the most interesting backstories of any molecule. So it's super interesting. Read it.
Yeah, and it's still on my bucket list to kind of make a pilgrimage to Rapa Nui. I feel like it's appropriate that I do that at some point. So anyways, it was studied for a while as an antifungal initially, and then as an anti-cancer drug, but it actually first got traction clinically in organ transplant patients as an immune suppressant. So that was actually what the drug was FDA approved for. This was more than 20 years ago now. For use in kidney transplant patients to prevent organ rejection. And then it got expanded, as is often the case, to other types of organ transplants. So, if you look at the clinical literature, or you just do a Google search, you will definitely find, rapamycin is an immunosuppressant. It goes by the clinical name of sirolimus. So people get kind of confused, but they're exactly the same molecule. That rapamycin or sirolimus is an immune suppressant used in organ transplant patients. Now, all of that's true. And it's sort of unfortunate that in some ways, it's sort of unfortunate that that's how the drug first was tested and approved clinically, because in that context, in sick people taking other immunosuppressants plus high doses of rapamycin, there are some side effects that go along with the drug. And so there's some concern in the clinical community about rapamycin and other mTOR inhibitors because of the side effects that are seen in organ transplant patients. They're not terrible. They're not typically life-threatening, but they're not pleasant, and there's some concern about them. What we're learning is that at lower doses, most of those side effects go away, and we can dive into that a little bit later if you're interested. But that's kind of the clinical story, and it's been used almost exclusively as in organ transplant patients, also for some forms of cancer, also for some diseases that are caused by mTOR hyperactivation, like tuberous sclerosis, but primarily organ transplant immune modulation. So sort of completely independent of that clinical use. Again, as I was saying before, around 2002 to 2004, about 3 different labs, I was involved in one, and then 2 in C. elegans, kind of hit on mTOR as a regulator of lifespan. So the development of rapamycin in the aging field was completely independent of the clinical work and its use in patients. We all were doing unbiased screens where we came across mTOR as a target for lifespan manipulation in these invertebrates. And then, of course, as soon as we saw mTOR, we did a PubMed search and found that rapamycin was a drug that inhibits mTOR, and thought, oh, maybe this drug will also affect lifespan. And it turns out it did. And, those 3 papers came out 2003, 2004. And we continued to work on mTOR and that pathway in the context of aging. But again, it really didn't, I think, capture the attention of the field until the interventions testing program tested rapamycin in mice, where they showed in 2009 that you could start treating mice at 20 months of age, which is about the mouse equivalent of a 60, 65-year-old person, and get lifespan extension. And that's kind of a funny story. I don't know if you guys know the story about why they started testing it at 20 months of age. It's kind of a funny story. I'm happy to tell it if you want me to.
Yeah, I think I have heard it before, but no, please go ahead, because no, I think it's
so first of all, just a little background on the interventions testing program. So this is an NIH funded program that's been around, since, I don't know, 2005, maybe 2004, where scientists in the field, or even the general public, can nominate interventions for testing on lifespan in mice. It goes through a review process. Anywhere from 3 to 7 are selected each year to be tested, usually small molecules, but it can be dietary interventions and things like that as well. The experiments are done at 3 different sites. So there's triplicate replication built in. They're done independently, and they're done in a genetically heterogeneous background called het3. And this is because there's a lot of concern about testing interventions in really inbred backgrounds like C57 Black 6. So, sometime in 2004, probably Rapamycin was nominated by a professor. His last name is Sharp. And he actually, I think, was inspired more by the effects of rapamycin on cancer that he was seeing than by the invertebrate work that I was talking about. Anyways, he nominated rapamycin to be tested. It was selected. And then they usually start the animals at about 6 months of age on the intervention, 6 or 9 months of age. It's changed a little bit, so young. But they found with rapamycin that they couldn't get it stable in the food. So the way they deliver all the interventions is in the diet. And they realized that the chemistry of rapamycin was such that it went bad if they just added it to the chow. But because they're testing multiple interventions at the same time, they have a large cohort of mice, all age matched, that they have to start the interventions at a specific point in time. And so the decision was made that, well, we don't think it'll take very long to figure out how to troubleshoot this. So we'll start the cohort, and then we'll just give them rapamycin as soon as we get it figured out how we can stabilize it in the food. It turns out it took them about a year to figure out how to stabilize it in the food. This was work that Randy Strong at San Antonio did to figure out an encapsulation method that would stabilize the rapamycin so it could be added to the chow. So by the time they actually had that figured out, the mice were, 17, 18 months old. And, the decision at that point had to be made. Should we actually do this? Should we try it? Or should we just wait for the next cohort? And I think probably almost everybody thought there's absolutely no way this is going to work starting at 20 months of age, because up until that time, nobody had ever shown that any intervention that extends lifespan in mice works robustly if you start at 20 months of age. Like, it was assumed that you had to start early. And this is actually a bigger point, because this is a way I think the field has fundamentally changed. Back in 2009, the dominant paradigm was that interventions slow aging, that we are slowing the damage that accumulates with aging. And so the expectation was, if you start after the damage has already accumulated, at best, you're going to get incremental effects. So nobody thought this was going to work, but they made the decision to go ahead and do it anyway. And thank God they did, because it worked. And so that, I think, it really was a paradigm shifting moment in the field, where suddenly it went from you have to start early to get a significant benefit to, oh, wow, we might actually be able to start in middle age and get a significant benefit for lifespan and potentially health span down the road. And that's really important from a translational perspective of really believing that you can have an impact in humans, right? Because we all recognize if we have to start treating teenagers with whatever our intervention is, that's going to be really hard, probably not feasible. But if you can start treating people in their 50s, 60s, maybe even 70s, you really have a chance to have an impact. So I think that was a real watershed moment in the field, the demonstration that an intervention could have a big impact starting in middle age. And now we know with rapamycin, at least, you don't even have to do it continuously for middle age. You can treat for 6 weeks or 3 months, still get pretty big effects. So that's the backstory. It was really an accident, right? And that happens in science sometimes. You have these fortuitous accidents that end up having a pretty big impact because you weren't doing what you expected to work. You found something that you didn't expect to work. So that's the story with rapamycin and the ITP. And this paradigm shift in the way we think about aging, I think, is really important. And we actually now have some biological understanding for how you can actually restore function, right? I think there's a lot of hesitancy, and deservedly so, to talk about reversing aging, because it really depends on how you define aging when you talk about reversing aging. But I think you can definitely say we can restore function and we can rejuvenate tissues and organs functionally, at least in mice. And there's, I think, pretty good evidence in people that we can, in some cases at least, rejuvenate function in tissues and organs. And so again, that's a really, really fundamentally important change in the way people are viewing aging biology. And I think it's informed the way we think about aging biology, and it's helped us understand mechanisms, because the mechanisms that drive aging, you have to fit them to the data. And the data says that you can restore function. You have to then have biological mechanisms that allow that. And I think that's helped us understand the aging process better.
I think you jogged my memory halfway through that. And I believe I think we had on Arlan Richardson, who gave us a really, just like your story there, really deep backstory, which I think just builds into this, the whole story around rapamycin, which is great. And coming towards the modern day, I mean, I've seen on Twitter recently, as early as I think last night, you were tweeting about the rapamycin study. So a little bit of a tangent from the dog aging project. But do you want to just give us a little bit of an overview of that as we're talking about rapamycin at the moment?
Yeah, sure. So, the study itself is again, it's observational. So it's not a randomized clinical trial. We're not treating people with rapamycin. Our specific study, which was funded by the Impetus Grants Program, I just want to give a shout out to the Impetus program. I think that has been hugely valuable to the field. These are relatively small grants, but they're funding a lot of projects that you would never be able to get funded through the NIH or through other governmental funding bodies. So I think it's been really, really important and valuable. So this is a project that was funded by the Impetus program. And the goal is really to try to collect information from people who are currently taking rapamycin or who have taken rapamycin in the past, try to understand what their experience has been, both from their any perceived benefits they think they might have gotten from the drug, and also any side effects. And we will have a control group of age and sex matched people who have never taken rapamycin to try to understand. I mean, if you think about side effects, first of all, are a really tricky problem when you're talking about older people, right? Just like in older dogs, right? I say this, in our trials, one of the reasons why we have a placebo group is if you've ever had an older dog, you will recognize that in any 3-month period, your dog is going to have one or more of this long list of things that include, throwing up, diarrhea, right? Lethargy. The same thing's true in old people, right? So, independent of what they're taking. So you really need to have an understanding of what the background frequency of some of these things that get called side effects in clinical trials is. In a clinical trial, you've got a placebo group. In people who have been taking rapamycin off label, for potential effects on aging, you don't have a placebo group. And so that's partly why we're trying to build that in. So anyways, it's only survey based right now. We are discussing with people who are interested in potentially trying to get samples from some of these patients, but ours is survey based and medical record and dental record review based to try to address, what are the experiences of people who have been taking rapamycin off label, not for organ transplant, but for, in many cases, the expectation that it might impact their health and longevity. This is actually a really interesting group of people, pretty mixed, really actually a lot of extremely successful people, in their professional lives, who are also very much into, wanting to maximize their healthy longevity. So CEOs of big companies, things like that. So, but also then you've got the biohackers who are interested in living longer, who are often taking lots of other things at the same time that they're taking rapamycin. So it's a complicated group of people. And so I think, we'll see what the data looks like, but the goal is really to try to at least get a feel for safety and maybe some hints about efficacy. And one of the reasons why I thought this was important to do gets back to, what I talked about before about the way rapamycin was developed as an organ transplant agent, or organ transplant anti-rejection agent, the side effects that go along with that, the reputation that rapamycin and other mTOR inhibitors have in the clinical world. That's driven largely by that patient population, which is fundamentally different than people in their 50s, 60s, 70s taking, rapamycin once a week for maybe health span promoting benefits. So, we don't have any real data on side effects in that, in the latter population. And so this is an effort to try to start to gather that. And hopefully, if we see that there aren't much in the way of side effects, change clinical opinion. So the idea would be to publish this someplace where clinicians can actually see it. I'm also hopeful that we'll identify a few interesting case reports. We've already got a couple of people who have had interesting experiences when they start taking rapamycin. Again, I think clinicians, they tend to be responsive to actual patient experiences, rightly so. And so I think that, maybe these case reports can also help to people to identify places where, rapamycin might be particularly useful from a clinical perspective. I just want to add as well, so that's our study. I'm very excited about the fact that there are going to be several clinical trials, it looks like, starting this year on rapamycin for specific age-related indications. So again, 2 of them funded by the Impetus program. One is a clinical trial for periodontal disease that Jonathan Ahn at the University of Washington is setting up. This is based on work that we did in my lab showing that we could reverse periodontal disease in old mice with rapamycin. So he's going to do a clinical trial in people. And then Zev Williams and Yushin Su at Columbia are pursuing a clinical trial for early ovarian failure in women at the Reproductive Health Center there for rapamycin. Again, based on mouse data that you can actually delay or partially reverse ovarian failure during normative aging in mice. And then there's a clinical trial that is being set up in New Zealand to look at muscle function and the impact of rapamycin on exercise. So those are 3 that I know about that I think, if all of those get off the ground and they're completed, I think, might give us some pretty interesting data on specific indications where rapamycin could have beneficial effects in the context of normative aging.
Definitely. Well, yeah, those are absolutely fascinating to hear. I didn't know about those 2 most recent studies that you're talking about. So yeah, I think I won't be the only person who's really looking forward to seeing some data come out of that. To go back to the dog aging trial, other than rapamycin, is there any other interventions that you're actually looking at in terms of dog aging?
Yeah, we're not currently. And it's funny because I must get an email once a month from somebody who wants me to test their particular intervention in dogs. And sometimes it's kind of crazy, but sometimes they're really good ideas, right? Like these things really should be tested. The fact is that it's a lot of work. That's the understated way of saying, what it takes to actually design a clinical trial like this and execute it, right? So just to give you a feel for the rapamycin study to have a statistical power to detect a 9% change in lifespan in dogs, it's a 3-year study with 580 dogs, right? So that's a big, for veterinary clinical trials, that's a big clinical trial. And so just figuring out the logistics of how you do that, where your clinical sites are, identifying the veterinarians, who want to participate. And I'll say veterinary medicine it's not like human medicine in the sense that there are a lot of clinical trialists, right? Most veterinary clinical trials have been relatively small, usually one center trials. And so, there's a lot of logistics that have gone into building this. I think it will be easier now. And one of the reasons, again, I recognized this early on, you don't know when you do a clinical trial whether it's going to work, right? And sometimes clinical trials can fail even if the intervention itself works for a variety of reasons. So recognizing that, I knew going in that one important thing we could do with Triad is build a template for how you do longevity clinical trials in dogs. And I think we've done that. I think we've created a model that other people can follow. I just don't have the bandwidth to do it right now. So the answer to your question is no, we're not testing other interventions, but I think there are a handful, that it makes sense to test in dogs right now. Again, because these are people's pets, you really have to think hard about safety, and you have to be sure, and you have to do the safety trials to make sure that it's very unlikely you're gonna hurt anybody's pet badly. I think of it almost like a pediatric clinical trial in that sense. People feel very similarly about their pets as they do about their children. So safety has to. So some of the things you might think about testing, because we can test them in mice, aren't going to be suitable for testing in dogs because of concerns about safety. But I still think there are probably, at least right now, we could come up with a set of 5 to 10 interventions where there's enough data that it probably makes sense to test them in dogs, maybe not for lifespan, but maybe for specific age-related indications.
Yeah, thank you very much, Matt. I will take over now with a few questions.
Sure.
And that probably is a quite tricky question to ask, but I will go ahead anyway. So overall, including and apart those interventions you just mentioned, what are, in your opinion, the major drivers and factors that would you do increase or decrease canine lifespan?
Right. So let me make sure I understand what you're asking. Are you asking like what interventions I think might have a big impact? Or are you asking what diseases are most important for canine longevity?
I think in a way both. So basically any kind of factors that we are aware of or that you think are
Sure.
main drivers to increase or decrease lifespan.
Okay. Yeah. So, I mean, I think, it's interesting because in some ways, it's very similar to human longevity in a general sense, right? So we know that obesity is an increasing problem in pets, and that obesity in pets leads to a variety of higher risk for diseases and negative outcomes during aging. We know that lack of exercise is a problem that can impact later health in life in pets. So I think those are, I mean, much like in people, those are probably the two of the most important factors that are also the most easily fixed, at least easily from a conceptual perspective, maybe not easy from a pragmatic or in practice perspective. But in many ways, it's very much like humans. It is the case that older dogs do show a somewhat different disease spectrum than people do. So, just like in people, cancers increase exponentially with age in dogs. So, but even more so. So cancer is probably the single most important lifespan limiting disease in dogs. So obviously, if you had a way to cure all cancers, you would expect that to have a significant impact on dog life expectancy. I would suggest that curing cancer is going to be just as difficult in dogs as it is in people. And by that, I mean pretty much impossible at the population level. I think this idea that we are going to ever completely eradicate all forms of cancer is just not based on what we know about biological reality right now. But I think you would expect it to have an impact on life expectancy in dogs. Dogs don't get a lot of vascular disease. So, things that impact vascular disease in humans probably are going to have a lower impact in dogs. They do get a lot of kidney disease, so interventions that would impact kidney disease. So again, I think it's very similar to people in a lot of ways. In terms of what can owners do, right now to have a positive impact on the health of their dogs, again, I think it comes back to what we already more or less know, right? Make sure your dog's not obese, exercise your dog regularly, get it vaccinated. I'm sorry to all the vaccine deniers out there, but it is a fact that vaccines help, at least for some things. Take your dog to the vet regularly, get a regular checkup. Oh, the one that almost everybody does not pay enough attention to is oral health. And this is true in dogs. It's also true in people. Brush your dog's teeth. I get why people are hesitant to take your dog in for a dental exam if it requires anesthesia. I admit I'm also hesitant to do that with my dog unless he needs to be under anesthesia for a different reason. But it's easy to brush your dog's teeth. It's really, really easy. Do it. It's sort of surprising how few people recognize how much periodontal disease and poor oral health increases in both people and dogs the risk for a variety of other age-related diseases. So there's a fundamental connection there that I believe is related to the biology of aging, that if you have poor oral health, you have higher systemic inflammation, which leads to dysfunction throughout the rest of the body or contributes to dysfunction throughout the rest of the body. So that's something that everybody can do that's pretty easy in dogs, and you should do it. And it's underappreciated. I have no idea if I actually answered the question you were asking, but hopefully it was good content regardless.
No, it definitely was. And you even did go a step further and almost completely answered my next question that I was going to ask, because I was going to ask what you consider to be the most promising strategies really to help improve the health span of our animal companions. And a part of those that you named, are there any maybe products or treatments that you can think of that might be very promising for the future?
Yeah, so I certainly wouldn't, I don't want to recommend any specific product. First of all, I'm not a veterinarian, so I'm careful not to recommend, specific commercial products. I feel okay saying brush your dog's teeth. I don't think I have to be a veterinarian to say that. But I don't want to recommend any products. I think, so I am encouraged that there is a growing interest among companies and academics in this area of, veterinary geroscience. I just made that up, but I think it's going to be a growing area of interest and importance going forward. This recognition that we have an opportunity to significantly impact healthy longevity in pets by targeting the biology of aging. And I've been very encouraged to see, a dramatic growth in people thinking about that. Now, I should say it's been a dramatic growth because for a long time, we were really the only ones at the Dog Aging Project thinking about it. So when you go from 1 to 10, that's a big growth. But I think it's exciting to see that there are people, including companies now, which is really great, right? There are some biotech companies and even some big companies that I've been contacted by who are looking at the biology of aging as a target for healthy longevity in pets, including, big multinational corporations that are the giant in anything to do with veterinary medicine. And I'm not going to name them, but anybody who knows anything about veterinary medicine knows who I'm talking about. They have a specific interest in aging as a target for health in veterinary medicine. So I think that's really great. And I think that, that will only accelerate the understanding of aging in companion animals and our ability to have an impact on health and longevity in companion animals through that understanding. And that may end up being drugs like rapamycin. It may end up being nutritional strategies. It may end up being other sorts of, environmental changes that can be made. And I think it'll probably actually end up being a combination of all of those things, as well as preventative measures, right? Or diagnostics. I think there's a place for all of those things to have an impact.
Yeah, thank you very much. And going back to the study frame and having dogs as an animal model per se, what would you consider to be the biggest challenges with working with dogs?
Yeah, so there are a few. I would say, one that I appreciated, and I already alluded to this going in, especially for a clinical trial, is the safety, right? Because these are very much like people's children. In fact, more than half of pet owners consider their pet to be part of their family, and they view them very much like a child. You really, really, really want to make sure you're not going to hurt anybody's dog. So that limits the kinds of interventions that you would ethically want to consider testing in companion animals. I'm not saying that's a bad thing. I actually think it's a good thing, but it is a little bit of a challenge because you really have to make sure before you start a large clinical trial that you can do this safely. So that's one. I think, and I also alluded to the fact that veterinary medicine, is pretty small compared to human medicine. There are not that many veterinarians at veterinary teaching hospitals with clinical trials experience who do that, as a big part of what of their career or their day-to-day experience. So you really have a small number of people that you can collaborate with to carry off these large clinical trials, which makes it more challenging. It's also hard because most veterinary clinics, private practice clinics, they don't have the same capabilities that a human medical clinic would have in a lot of ways. And they're really, really busy, right? These veterinarians are going from patient to patient to patient to patient. They don't have time really to participate in a scientific study. So, you have to figure out ways to, first of all, identify those veterinarians who really want to participate, but also continue to engage them in the process. And that's, again, it's easier to do that in human medicine because you have physicians who they specialize in participating in scientific studies or in clinical trials. So that's been a little bit of a challenge. I think, we'll find out. We've been very fortunate that it hasn't been so hard to recruit dogs into the overall dog aging project. We'd obviously love to have more, but we've got about 37,000 dogs in the longitudinal study right now. And I think that, again, just reflects the fact that, people love their dogs and they like to talk about their dogs and they're excited about participating in a scientific project, that's about their dogs and might have an impact on their dog. So that part hasn't been hard. We will see as we go forward how challenging it is to retain participation. Recruiting hasn't been a huge challenge. Retention is a little bit of an unknown. So we'll see. The other thing I should say is I think, our unique experience with the Dog Aging Project, has of course happened during a global pandemic, right? So, some of the challenges around actually building the clinical trial and getting the veterinarians on board and getting the clinic set up to do the study, obviously that has been impacted by, lockdowns and emergency procedures only and things like that. So I'm hopeful that as the world hopefully comes out of this global pandemic, that, you know, things will get easier in that sense, and people won't be so overwhelmed with just trying to get through the day. So I think our experience may not be completely normal in that sense going forward.
All right. Yeah, thank you very much. I think that gives us a good insight into the hurdles you may come across running such a project. And yeah, one big program in our field and the biomedical sciences in general is the unsuccessful translation from establishing effective intervention in animal models to the same intervention being effective in humans.
Yeah.
So what makes dogs a better and unique model in which to study aging when compared to classic models?
Sure. So first of all, let me take a step back, because I think in some ways this is a little bit of a myth. The idea that animal models, you know, have a terrible record of translating through to humans. First of all, in the context of aging, we have no data, right? There is no data on an intervention that worked in an animal model and failed in humans. That doesn't mean that when we actually do the trials that won't happen, but we don't have any data. So it's an N of 0 there. So we can't say anything about that. But, you know, people often point to cancer as an example. We've cured cancer 1 million times in mice and it didn't work in people. And that's kind of true. But I actually would suggest that maybe the major reason why the animal models have failed to be, you know, as good at translation as we would hope is not because of the animals, the mice themselves. It's because of the flawed way that the biomedical community has used them. And in particular, I'm referring to the fact that most of the diseases where the animal models have failed miserably are cases where it's an age-associated disease, and people have tried to artificially create the disease in a young animal. And this is a frustration point for me, and that I'm trying to communicate to the non-geroscientists in the world. And it seems like it should be obvious. The physiology of a young mouse or a young person is fundamentally different than the physiology of an old mouse or an old person. And so when you take a disease that in the real world happens in the physiology of an old person, and you try to model it in a young mouse, it's not shocking that that is not a very good model. So all of that is to say, I'm not sure it's only or even primarily the mouse to human problem here. I think it's the fact that most of these people have been studying diseases of aging in a young physiology, and it's just completely different. So I don't accept yet the premise that mice are not a good model for X when you have used the mice improperly from an experimental perspective. Okay, so that's my tirade on that. So why are dogs better? So again, I don't know if they're better, but I think they should reflect the human situation more than a laboratory mouse. So one is, you know, they share our environment. We've already talked about that. They're genetically diverse. We've already talked about that. And you can, in fact, study these diseases or functional declines in the real world in an old animal that's experiencing the real world, right? So, and you can do it in a reasonable time frame. So I think that's the real power from a biological perspective of dogs as potentially a better model for diseases of human aging or functional declines of human aging. But, you know, I think, again, we do have to admit that that's all intuitive and it makes sense. We don't actually know that dogs are going to be a better model for human aging. I believe they probably will for those reasons, but, you know, time will tell. We have to get the data and we have to actually find out. Everything that I know, you know, and this comes in part from having old dogs. I've had several old dogs in my life. They get old very much like we do. And I've known several old people in my life, as I'm sure you all have, right? Old dogs show a lot of the same functional changes, behavioral changes that old humans do. And so they look like, just from the phenotypic output, it looks a lot like human aging. And again, I think we can see that more in dogs, obviously, than we can in laboratory mice. We can see a greater diversity of phenotypes of aging in dogs than we can in laboratory mice. So I feel pretty good about the idea that at a cellular, molecular, functional, phenotypic, at all of those levels, there are aspects of aging that are very highly shared between dogs and people.
Right, yeah. And are there any specific implication that we can maybe at this stage already draw from the findings that we gather from research with dogs as an animal model to human longevity?
So I don't know that there's anything I feel, 100% confident about. Honestly, I don't know that there, first of all, I would say there's not a lot about human longevity that we should feel 100% confident about, right? I think it's a matter of, growing information and data availability and then kind of making the best guess you can at that point, right? So what I would say is, I've already talked about, I think we can be pretty sure that just like in people, relationship between obesity and health outcomes and lifespan is pretty similar. Relationship between exercise and those things is pretty similar. In terms of, specifically what we've learned from the dog aging project, I don't know that there's anything I feel 100% confident about. It seems like with rapamycin, just from our 2 safety trials, it does seem to be the case in both of those trials, only one of which has been published. In both of those trials, the owners self-reported that the dogs getting rapamycin were more active, and the owners didn't know if their dogs were getting placebo or rapamycin. So, that suggests to me one possibility, and this is anecdotally what people have reported in older people taking rapamycin. One possibility is that, the effects of rapamycin on systemic inflammation are reducing chronic pain in old dogs, and that's observed as increased activity. Again, that's very speculative and it's not quantitative, but the fact that it came out of both studies, owner reported without us even specifically asking about activity, makes me think it's probably real. So, I'm being careful to say that's what I think. I don't know, but I think that, it gives me a fair amount of confidence. Like if you asked me what my prediction is for rapamycin in humans, I would say I'm pretty confident that rapamycin has benefits in healthy older people for at least some aspects of biological aging. And in particular, from my own personal experience and from people I know, it seems to be really pretty effective at age-related inflammatory conditions in joints and capsules, right? And so it would not shock me if there are benefits for things like arthritis or, inflammation of capsule, like shoulder capsule, which was my experience, and the pain that goes along with it. So, again, I can't prove it. I haven't done a clinical trial, but I feel pretty personally confident that that's probably the case.
Great, thank you very much. So dogs are often called man's best friend. And what impact do you think it will have on the general population if the dog aging project is successful and we're able to significantly increase lifespan of humanity's greatest companion?
Yeah, right. I mean, I think that's certainly something that I thought a lot about, when first embarking on the dog aging project is, the potential for this project to change the way that a lot of people think about aging, right? So I already alluded to the idea that I think dogs are actually, really valuable examples for just the fact that aging rate is different between different animals, right? 7 dog years, 1 human year. So I think that's powerful. But of course, if we get to the point, let's say 4 years from now, when we unblind Triad and we look at the data, where we can say that in a large clinical trial, rapamycin slows aging in dogs. And by that, I mean significantly increases lifespan and significantly reduces functional declines or diseases of aging. If that's the outcome of that trial, yes, I think that could have a big impact on public perception of the field of geroscience and also clinical practice, right? Again, obviously, people aren't going to immediately assume that because it works in dogs, it works in humans, but some will. And certainly I could see many veterinarians becoming comfortable with prescribing rapamycin at that point, especially if the safety data look, the way we expect. They're really going to be very little in the way of side effects. So yeah, I think it could have a big impact from clinical perspective. And then of course, for those people who consider their pet, their companion animal to be part of their family, if you can say we have a way to give a family member extra healthy years, that's a big deal that will resonate, I think, with a lot of the general public. So again, I try not to get too far ahead of myself because, as I said, we don't know the clinical trial is going to work, right? It's quite possible that we'll get to the end of the trial and have, it'll be suggestive, but maybe not, definitive, right? So I don't want to count on that happening. But I think if it is definitive, then it will have a big impact. I'm hopeful that independent of the clinical trial, though, that the dog aging project itself can help expand the recognition and understanding among the general public of aging as a biological process, of the geroscience paradigm, and that there's actually progress being made in this field. I think we all recognize that there's a constant battle and push and pull with the snake oil and the hype and the science fiction side of the equation, where sometimes it seems very challenging for the rigorous, legitimate scientists to have a chance against that stuff. But I think this is a case where we might be able to have an impact on public perception and help people to understand that, yeah, sure, there's a lot of noise out there, there's a lot of nonsense, but there's also real science and real progress being made here, and that we actually have the potential to have an impact on aging biology. And it's not a huge leap. Once they accept that that's possible in their pets, it's not a huge conceptual leap for them to accept that it's possible in their friends and family members and in themselves. So I think it can have an impact there as well.
Yeah, thank you very much.
Yeah, so Matt, you've been extremely gracious with your time. I realize we've gone well over the hour. And, between the three of us, I think we could keep you here for days on end asking you questions. Just if you've got a tiny bit of time left, I wanted just to jump over to Jared, just to ask if, Jared, you've got any quick questions you want to ask at the end. I know you've been waiting in the wings.
Oh, I mean, it's unfair to tempt me. Okay, can I do one more? So the translation, obviously, from rapamycin or fasting studies in like rodents to humans, we've already spoken a bit about that. And I like your point about the fact that we haven't proven yet that they don't translate. But obviously, the fasting protocol for rodents, the fast equivalent in humans is a much longer fast. Right? It's like a 15, 20-day fast. Do you think that rapamycin is like qualitatively a different thing, even though they're both acting somehow on mTOR?
Yeah,
that's
I think that's a really important question. So I think it's important to also recognize, even in dogs, the relationship between metabolism and caloric restriction or fasting, particularly fasting, the length of the fast and its effect on metabolism is fundamentally different in mice and dogs and people, right? So in dogs, at least this is my understanding, they don't go into ketogenesis for several days when they're fasting, right? Whereas people, often will go into ketogenesis within 24 or 36 hours, right? So it's different there too. It's different than mice and it's different than people. So this question of, is rapamycin somehow, might it be a similar sort of paradigm where, same dosing or same duration of rapamycin in a mouse is different than a dog, is different than a person. Again, we have to be honest, we don't know the answer to that. I'm encouraged by both our 10-week study in dogs and the 6-month study where we saw the activity of reported activity effects. And then in 10 weeks, we saw effects on cardiac function. That gives me some encouragement that shorter time periods in dogs with rapamycin, comparable to what we do in mice, which, the transient treatments with rapamycin in mice are usually 8 weeks to 3 months. And you see big effects on, periodontal disease, reversal of periodontal disease, reversal of heart function, restoration of immune function, improvement in cognitive function, all in that sort of 8 to 12 week period. So that gives me some reason to think that it might work the same way in dogs in that time frame. And then similarly, the 2 clinical trials from Joan Manick's group with everolimus, the rapamycin derivative, where they treated for 6 weeks, and they found some evidence for improvement in response to a flu vaccine. This is in healthy older people. Also, suggestive that similar timeframes in people might be responsive to rapamycin. And again, I've sort of alluded to this. I've talked about it on Peter Attia's podcast. My personal experience with frozen shoulder was that in 10 weeks, I had basically a complete reversal, complete cure of frozen shoulder, 10 weeks of rapamycin. So I feel relatively confident that we can get some of the effects on inflammation, at least in that 10-week period. I don't know whether or not the other sorts of age-related outcomes will require a fundamentally different time period. This is really one of the challenges, right? With any intervention, you want to think about testing in a clinical trial in dogs or in people. There are so many variations on dose and duration and patient population and all that. You have to try to take your best guess at where you're going to see a positive signal and then go for it, because you could spend your entire life trying to game the system and come up with a completely optimal strategy. And you can't test everything, right? So that's kind of what gives me hope that that's the case. I also want to make a comment though, because there's a lot of confusion in the field about fasting. If you actually look at the mouse data, the evidence in mice that fasting increases lifespan is really, really poor. There's very little. There's one study that I'm aware of from Rafa DeCabo's lab where they mostly controlled for caloric consumption, and they get about a 9% increase in lifespan in that study. Almost all the other studies on fasting mimicking diet or intermittent fasting, the mice are calorically restricted. So it's not a true study of the effects of fasting. It's a study of caloric restriction. The other thing I'll say about that one study I just mentioned where they got a small increase in lifespan, the controls were pretty short-lived in that study. And so, it's a question whether if you did that same study and you really carefully controlled for caloric consumption in a longer lived population where the controls, were as long lived as they normally are, would you even get a 9 or 10% increase in lifespan? So there's this perception, oh yeah, fasting slows aging and increases lifespan in mice. And a lot of people share that, but if you actually look at the data, it's not so convincing that fasting is a robust way to increase lifespan in mice. I think you can get some small effects, but it's nowhere near as effective, at least so far, as rapamycin or true caloric restriction.
Fascinating. Well, yeah, Matt, thank you again. Finally, where can people actually keep up with your work with the dog aging project and also the rapamycin studies? Yeah,
so the Dog Aging Project website is dogagingproject.org, and we are absolutely still recruiting dogs into both the longitudinal study and the rapamycin clinical trial. Particularly for the longitudinal study, we would like to have more young dogs and puppies. So if anybody has a dog that you're interested in participating in the study, please go to the website. It's a really easy nomination process, and join the Dog Aging Project pack. For now, it's restricted to the United States, but I'm hopeful that at some point in the relatively near future, we'll be able to open it up to dogs internationally. And then for our human study, the website is rapamycinstudy.org. And we are absolutely interested in obtaining data from anybody who has ever used rapamycin. And as I mentioned, we are also going to have a sex and age matched control group of people who've never used rapamycin. So certainly if people are interested in participating in that study, I would encourage you to go to the website. It's pretty easy. It's a series of about 8 survey modules. They each only take 3 or 4 minutes. So it's pretty quick. And definitely we would love to have people participate.
Brilliant. Well, again, thank you for that. And yeah, guys, go and check that out. Matt, you've been so gracious with your time. Thanks again. It really is, it's a pleasure having you on. And yeah, it's a great privilege to me as well. And yeah, I would love to do this again, because like I said, I'm sure Jared and Antonia and myself, we've got 100 more questions we could launch at you. So maybe sometime in the future, we can do this again.
Great. Thanks a lot. It's been fun.
Yeah, thank you very much. And thank you for Antonia and Jared as well. And guys, yeah, we'll have this up on our YouTube channel, and we'll do the full transcription as well on our website. So yeah, thank you for joining, and we'll see you again next time. Goodbye.
Thanks, Chris.